DEPArray technology is the only technology on the market that combines microfluidics, microelectronics, and microscopy to isolate and identify cells at single-cell resolution. This is elegantly accomplished by loading your sample into our single-use microfluidic cartridge with integrated electronic electrode array. The microfluidics gently, with low pressure and no stress, inject the cells into the electrode array at the center of the cartridge. The electrode array traps the cells in a manner I will describe in subsequent slides and allows analysis of the trapped cells in our cell browser software at single-cell resolution. The diameter of cells in your sample that can be trapped in the chip range between 7 and 40 microns, which spans the diameter of the majority of cells that you will want to study. The microscope, which is one of the most important features of the DEPArray system, is embedded within the DEPArray machine itself. The microscope has optical magnification capabilities of both 10 and 20 power and can identify cells using multiple fluorescent dye channels and bright field illumination. The ability to characterize target cells prior to isolation and before downstream analysis saves time and money, allowing you to only analyze the cells that you want. This is in stark contrast to other microfluidic cell isolation methods, such as DropSeq, which characterize recovered cells only after isolation and sequencing based on the statistical probability of the cell's identity. On this slide is a brief but far from exhaustive list of all the different types of samples that have been run successfully on the DEPArray system. The take-home message of this slide is that if your primary source of sample can be made into a single-cell solution and it can be identified with a fluorescent marker, it can be loaded and analyzed on the DEPArray. How exactly are cells isolated and recovered from the DEPArray cartridge? Briefly, a suspension of fluorescently labeled cells is injected into the DEPArray cartridge. The array of electrodes at the center of the cartridge are then activated to form depth cages, trapping the labeled cells. The cartridge is then scanned in each of the desired fluorescent channels, plus under bright field illumination, and this information is compiled in our cell browser software. Using this software, the user then identifies target cells after analysis of multiple parameters, including fluorescent and morphological measurements, as well as visual observation of image cells. In the following step, the depth cages are automatically programmed to move the target cells to a parking area from which individual cells or groups of cells can be recovered and dispensed into the collection vessel of choice, be that a PCR tube for downstream genomic analysis of a 96-well plate for cell culture. How are the depth cages formed and how do they actually work? The electronic array in the center of the microfluidic cartridge is comprised of electrodes whose polarity is controlled by software. If the electrode array consisted only of positive electrodes, when your sample is injected into the cartridge, the cells would migrate around and settle randomly within the array. However, using software, we can create dielectric phoretic or depth cages, hence the name DEPArray, by surrounding a negative electrode with positive electrodes as shown in the top left corner of the schematic, here indicated by the red rectangle. When a uniform electric field is applied to the electrode array, a minimum of the electric field is formed above the negative electrode and the cell is trapped in this area. To better understand how the depth cage is formed, this is a cross-section of the electrode array. Again, the depth cage is formed where the minimum of the electric field exists. The white arrows represent the direction of the electric field. Just like when running an agarose gel, the electric field flows from positive electrode to negative electrode. The top of the array is negatively charged, so the electric field flows toward the top of the array and then is drawn down towards the bottom of the array by the negative charge. When the cells are injected into the array, they follow the electric field and are also drawn towards the negative charge and trapped. To move the cells around the array, the polarity of the electrode is simply changed, and the cell is dragged by the change in the minimum of the electric field to where the new depth cage is formed. This continues until a cell encounters an obstacle, in this case, a cell trapped in another depth cage. The software recognizes that the cell is an obstacle and does not allow the cell that is moving to migrate close enough to the other cell for them to accidentally interact. The cell that we want to move is simply directed around the obstacle at a safe distance. To give you a better idea of the DEPArray workflow following cell trapping in the depth cages, I will now show you a brief video. Once the cartridge has been loaded into the DEPArray and the sample has been injected into the electrode array, the microscope scans the array in the fluorescent channels your sample is stained with and in bright field. You can watch and analyze the chip scan live and alter characteristics of each fluorescent channel, such as exposure, gain, and focus, in order to obtain the optimal images and calculations for each channel. Once the chip scan is complete, The cell browser software assigns each identified cell a unique ID, calculates the fluorescent characteristics for each cell in each of the channels that were scanned, and creates an image gallery of the scanned images for every cell shown at the bottom of the software. The researcher then uses this information to decide what cells to recover for downstream analysis. This can be done using histogram analysis mode, not shown, or scatterplot analysis mode as shown in this video. This is identical to gating cells in flow cytometry experiments. You plot one fluorescent characteristic against another characteristic and gate the population that you are interested in recovering. You can also visualize where the gated cell population is located within the electrode array and where the gated population is located in relation to all other cells identified during the chip scan. After this, the gated population can be refined by analyzing the images in the cell gallery. Here, you can determine if each cell identified during the scan is indeed a single cell, or if it is more than one cell, something that is impossible with flow cytometry. You then group the cells that you want to recover in recovery groups and move to the Recovery Manager mode. Here, you determine your recovery scheme. You can recover a single cell in the first well, a group of cells in the second well, and so on. Once you have determined your recovery setup, you tell the software to move the selected cells from the main chamber to the parking chamber. This step can also be watched live using the live camera window in the upper right corner. This is a real-time view of cells moving within the array. When the cells are moved into the parking chamber, they are aligned in columns ready to be recovered. The software automatically calculates a route for the cell or cells to move from the parking chamber to a specific spot in the recovery chamber. Once the cell has reached this spot, the cell is dispensed into your collection vessel of choice. The recovered cells are dropped into the collection vessel through gravitational force, and you can watch the drop form using the live camera view. The recovered cells are now ready for downstream analysis. The DEPArray technology was originally developed to isolate very rare cell populations from a heterogeneous background, one of the best examples of this being circulating tumor cells, CTCs. The difficulty with isolating CTCs is that they can be extremely rare. Depending on the tumor being studied, they can occur with a frequency of only one cell in 1 billion cells. Many different methods currently exist to enrich CTCs, but these methods don't purify the CTCs. They just reduce the amount of background or non-target cells in your sample. Using DEPArray technology, you can isolate single CTCs to study patient tumor heterogeneity on a cell-by-cell basis, or you can isolate a group of pure CTCs for bulk downstream analysis of the entire population of CTCs. 10 papers have been published over the past 2.5 years concerning molecular and functional characterization of CTCs isolated from the DEPArray. The most prominent DEPArray publication about isolation of CTCs comes from Caroline Dives' group at the University of Manchester in the UK. This publication highlights a very important aspect of cell isolation using the DEPArray, and that is the fact that the cells isolated from the DEPArray are viable and able to proliferate after recovery. This is illustrated by the ability of DEPArray isolated CTCs to form CTC-derived xenografts or CDXs that recapitulate the genetic profile of the population of CTCs used to generate the xenograft and even the drug response profile of the patient. Once the rare cell population workflow was established, we turn to another important facet of cancer research, and that is the heterogeneous nature of tumor samples. To tackle this difficult problem, we determined to use our technology to resolve the heterogeneity and small sample size difficulties that arise when working with FFPE samples, although this workflow in a modified form can also be used to resolve the heterogeneity of fresh tissue biopsies as well. As you all know, an FFPE tissue sample is typically deparaffinized and DNA is extracted from all the cells contained within the tissue section, including both tumor and non-tumor cells. This makes downstream genomic analysis complicated, or in some instances, impossible, depending on the percentage of tumor cells within the FFPE sample of interest and the size of the FFPE sample. DEPArray technology provides a solution to this challenge by allowing you to collect only tumor cells and only stromal cells from the same FFPE sample. Importantly, this also alleviates the need for a matched normal control sample, which in the case of old archived samples may not even be possible to obtain. This is an example of what a dissociated and antibody-stained FFPE sample looks like in scatterplot analysis mode on the DEPArray. The x-axis of the scatterplot is the keratin stain. This identifies tumor cells of epithelial origin, and the y-axis is the vimentin stain. This identifies stromal cells. The upper right quadrant of the scatter plot contains double positive cells. Using the cell image gallery, we can recognize that some of the points in the double positive quadrant are actually two cells in the same cage, one cell that is keratin positive and one cell that is vimentin positive. But interestingly, there are also cells found in this quadrant that are truly double positive, and this could indicate that these cells are undergoing EMT, epithelial mesenchymal transition. The lower left quadrant, the double negative cell population, is probably comprised of stem cells and infiltrating tumor lymphocytes, or TILs. TILs are a very interesting cell population of cells to study, especially in a therapeutic setting. And to this end, Silicon is currently developing a protocol to isolate TILs from FFPE samples. To further confirm that the gated keratin-positive cells are actually tumor cells, a parameter termed integral intensity DAPI can be calculated. Integral intensity DAPI is calculated by normalizing the fluorescence intensity of light emitted from the DAPI channel to the diameter of the nucleus, returning the total amount of DAPI signal in a single nucleus. This allows clear-cut determination of whether a cell is diploid or hyperdiploid, as shown in the schematic on the right. The stromal cells, graphed in the bottom histogram, have a characteristic peak of diploid cells, 2N DNA content, and a much smaller peak of cells with 4N DNA content, most likely cells in S phase. In contrast, the tumor cell population in the top histogram contains almost no cells that correspond to the diploid peak from the stromal cell population, with the majority of these cells corresponding to the 4N or greater DNA content area of the histogram. We recently published a paper in Nature Scientific Reports outlining our newly developed FFPE workflow. In this paper, we dissociated and stained FFPE samples from several tumor types with varying lengths of storage time from 1 year to as long as 21 years. These samples had varying amounts of tumor cellularity as determined by a pathologist, ranging from as low as 5% tumor to as high as 60%. This is an image of the lung FFPE sample with only 5% tumor cellularity, of which we were able to successfully recover pure tumor cells using the DEPArray. I'm often asked what size of FFPE sample you need to process to have enough cells to run on the DEPArray. This slide illustrates FFPE samples that have been prepared for analysis on the DEPArray. The sample on the top and in the middle was probably just a tissue slice, while the sample at the bottom was the entire FFPE sample. I want to emphasize here that we only need a small number of cells to recover your target cell population of interest. Of course, this is dependent on how abundant your target cells are in the sample. However, we can even recover tumor cells from fine needle aspirates, which can contain only a few 10,000s of cells, even though the vast majority of these cells are non-tumor, non-target cells. Now that you've isolated your rare single cells or your population of pure cells from the DEPArray, what do? We have developed several downstream genomic solutions, and these applications are what I will focus on during the rest of my talk. However, it's important to remind you and to emphasize that we can isolate live cells that are viable and capable of proliferation. So you could, for example, isolate a single cell or a population of cells to develop and characterize cell lines derived from a single cell, or recover live cells for RNA analysis. For downstream genomic solutions, we have initially focused on DNA, and the first thing that you need to do before downstream DNA analysis of your recovered cells is to amplify the genome of your isolated cells. We have developed a WGA kit that is specifically designed to address the challenges of amplifying DNA from a single cell, but can also be used with up to 1,000 cells per reaction. Our genome amplification kit is a one-tube, one-day procedure. We can produce up to 2 micrograms of double-strand DNA per single cell, which can be used directly in PCR without prior purification or dilution. Alternatively, the WGA product can be re-amplified with our Ampli-1 Re-Amp kit to produce an almost endless supply of DNA from the initial WGA reaction. What sets us apart from other WGA kits is the deterministic and non-random method of amplification that we employ. With random primer-based amplification kits, it is very easy to have sequence representation bias, which generates a high rate of allelic dropout or preferential amplification of highly polymorphic sequences. These effects are more pronounced in samples with low DNA quantity, for instance single cells, or in samples with fragmented DNA template, for instance FFPE. One reason why allelic dropout can be problematic is when PCR results could lead you to believe that the cell you are analyzing has loss of heterozygosity, but this LOH is actually due to an artifact of your amplification method. If the LL you are analyzing is, for instance, BRCA1, this could at best lead you down the wrong research path and at worst lead to poor diagnostic decision-making. By utilizing a non-random PCR amplification method, a constant distribution of priming events across each allele and the entire genome occurs, greatly reducing the allelic dropout rate and preferential amplification problems of other amplification methods. This results in a high DNA yield, and importantly, this method is not dependent on the quantity or quality of starting DNA material. The allele call rate seen at the single-cell level is representative of the actual allele call rate of the starting sample. To perform this non-random deterministic WGA, we first lyse the cell or cells and fragment the genome with a restriction enzyme, MSE1, that has a short 4-BP-TTA restriction motif that is scattered approximately 19 million times throughout the human genome. The average spacing of the restriction site is 126 base pairs, making the resulting fragments ideal for subsequent PCR-based amplification. Following restriction digestion, PCR adapter sequences are ligated to the MSE1 sticky ends of the genome fragments. A 3' end filling PCR reaction is performed to complement the ligated sequence, and finally, WGA amplification is accomplished with a single primer pair that is specific to the ligated adapter. This ensures a reproducible priming pattern with increased PCR efficiency and high genome coverage. These characteristics are essential for analysis of clinical samples where only high fidelity to the original sample is acceptable. Using our WGA kit, you should expect 74% genome coverage with an allelic dropout rate of only 2%, compared to an allelic dropout rate of up to 60% using kits based on random priming amplification. The last question to ask before moving on to analyzing the amplified genome is, did the amplification actually work? There are several factors that can influence the efficiency of WGA that in many cases cannot be controlled by the researcher, such as the quality of cells isolated from a clinical sample. To save both money and time, we have developed a QC kit to check the quality of your amplified WGA products. The kit consists of a multiplex PCR reaction that covers 4 amplicons of different sizes and locations throughout the genome. The result of the PCR is between 1 and 4 bands, depending on the efficiency of amplification of a given sample. The PCR products named in descending order, D, C, B, and A, are a proxy for amplification of longer WGA amplicons in the case of D, C, and B, and shorter amplicons represented by band A. The longer amplicons represent the length of approximately 70% of the WGA fragments produced with the Ampli-1 WGA kit. We have created a Genome Integrity Index, or GII, valued from 0 to 1 that evaluates what downstream application can be performed on the WGA products. A GII of 0 means that the WGA was not successful. A GII of 1 to 4 was suitable for downstream PCR and/or sequencing, and a GII of 4 is suitable for a CGH or low-pass genome analysis. The presence of 3 to 4 PCR bands is predictive with 94% sensitivity and 94% specificity of successful WGA. Now that you have a successful WGA, what can you do with this sample? One possible downstream application is to analyze copy number variation. Traditional methods to analyze CNV, such as ACGH, are not ideal for samples with a small quantity of DNA or degraded DNA, like that which can be obtained from clinical samples or FFPE tissue. As an alternative to ACGH, we have developed a kit to analyze your sample using low-pass genome analysis, which is a digital version of ACGH. With this kit, you can barcode and pool multiple WGA products, allowing you to multiplex your sequencing reactions to reduce cost per sample. ACGH can cost about $250 per sample, whereas low-pass genome analysis, depending on the number of samples you multiplex and which sequencing platform, Ion Torrent or Illumina that you utilize, can cost between $50 to $100 per sample. What is low-pass genome, and what do low-pass genome results look like? Low-pass genome analysis is a method to assess copy number variation developed by a research group in the Netherlands. This group created a freely available software program to analyze data points generated from shallow whole genome sequencing. Sequencing coverage is only a 0.1x with an average read count of approximately 6 million reads after alignment to the human reference genome and filtering out of duplicate PCR reads. However, this coverage is sufficient for CNV analysis using this freely available software. The aligned genome is then divided into non-overlapping fixed-size bins, in this case 100 kb in size, which are used to determine copy number estimates by counting the number of reads in each bin. On this slide, you are looking at an unsorted bulk processed FFPE sample with 40% tumor content, as assessed by a pathologist in the top graph and DEPArray sorted stromal cells in the bottom graph. Each line within the graph represents a chromosome, and each data point represents a read count. This data is median normalized log2 transformed so that different samples can be compared to one another. As you can see, with unsorted cells, it is difficult to tell how significant the low-level changes in this sample are because of the high percentage, 60% of the original sample, of contaminating non-tumor cells which have no copy number change at all. This is in stark contrast to a CNV plot of pure tumor cells from this sample where CNV, both loss and gain, are immediately apparent. These results really speak for themselves, but I would like to point out the utility of this type of pure population or pure cell type analysis in both the clinical research and clinical diagnostic laboratory setting, especially in the area of clinical disease management and genetic counseling. Obviously, the more unambiguous the results of these analyses can be, the more certain you can be in your diagnosis or recommendation. The next application is our Modified Cancer Hotspot Panel, another application that could be a very useful clinical decision-making tool. Traditional NGS analysis of tumor samples results in the given frequency of a mutated allele in a sample, but will not tell you what the genetic makeup of the tumor you are analyzing is. For example, if your results are that a given mutant allele exists in a population of cells at 43%, you do not know if the sample contains a mixture of cells that are homozygous wild-type and homozygous mutant, a mixture of cells that are homozygous wild-type and heterozygous mutant, or a mixture of some cells that have LOH of the wild-type allele along with a mutation in the other allele, or if the cells have copy number variation. With the power of pure tumor cell populations versus pure stromal cell populations and the Cancer Hotspot Panel, this question can be answered. The Ampli-1 NGS Hotspot Panel is a modified version of the ION AmpliSeq CHP that surveys hotspot regions in 50-plus genes that are prone to mutation in many tumor types, as well as the unique addition of CNV amplicons for 7 genes known to have CNV changes in multiple tumor entities. This slide is an example of the results of the Ampli-1 CHP run on a lung tumor FFPE sample and presented at AGBT in 2015. Of the 50-plus genes in the hotspot, this tumor had mutations in 17 of those genes. The numbers represented in the table are of the frequency of the mutated nucleotide as compared to the human reference genome nucleotide call at the same position. The gray samples are from DNA isolated from the bulk unsorted FFPE lung sample. The green samples are pure tumor cells isolated on the DEPArray. The red samples are pure stromal cells. Each column represents a biological replicate isolated from the same sample. To save time, I will just use 2 genes as examples of the power of this type of analysis. Looking at the bulk unsorted results from the SMAD4 gene, I would assume that the tumor cells in this sample have a mutation in one allele and increased copy number of the wild-type allele. But when I examine the stromal-only population, I can tell that this patient is actually germline heterozygous for this particular gene, because the frequency of mutant allele is approximately 50%. Interestingly, from a therapeutic point of view, it turns out that the tumor has 0% frequency of the mutation. From these results, we can conclude that the SMAD4 mutation is probably not driving the growth of this tumor. A second mutated gene in this sample, STIC11, is actually known to be mutated in non-small cell lung carcinomas and can be a driver mutation in this setting. When looking at the bulk population, it looks like the tumor is heterozygous for this gene. Examining stromal only, the patient is homozygous wild-type, and examining the tumor, the patient is homozygous mutant. From these results, we can conclude that the tumor contains both the variant mutant and an LOH, because the probability that the same point mutation occurred on both alleles is very low. This is also a very good indicator that this tumor would respond to therapy directed against STIC11. Lastly, we can get very meaningful whole exome sequencing information from pure populations of cells. This is a map of single nucleotide polymorphism variant frequency across the entire exome. Approximately 54,000 variants were found in this particular FFPE sample. The stromal cell SNP frequencies are plotted on the x-axis, while the tumor cell SNP frequencies are plotted on the y-axis. The cloud of frequencies in the middle of the plot represent SNPs that are represented at almost the same frequencies in both the tumor and stromal cell populations. The SNPs at the very top of the graph are represented 100% in the tumor cells, and those at the bottom of the graph are at 0% in the tumor but at varying frequencies in the stromal cell population. These SNPs are interesting because they represent loss of heterozygosity in the tumor and could be potential druggable target variants. Those frequencies on the far left of the graph are somatic variants annotated in the COSMIC database and are randomly distributed across the genome. We can infer very interesting results about CNV and LOH when these same whole exome SNP frequencies are plotted using a B-allele plot. A B-allele plot plots whether the SNP is called as either homozygous wild-type 100, homozygous mutant 0, or heterozygous 50. When you examine the B-allele plot of unsorted cells, there is no conclusion that you can make because there is too much noise and background data points. This is because the SNP frequencies from the somatic-only cell population plot more or less right around 50, meaning these SNPs are generally heterozygous for these alleles. In contrast, regions of LOH and CNV are clearly visible in the tumor-only population. Highlighted in yellow are regions of LOH, where the SNP call is either 100% homozygous mutant, top of graph, or 0% mutant homozygous wild-type at that position. Highlighted in green are regions of CNV, where one allele is called more times than the other allele. The areas where the allele calls concentrate around 50% are balanced region of the exomes within that particular chromosome. The last calculation we can make with the whole exome data is a determination of whole exome copy number variation. When looking at a B allele plot, we can tell that the allele copy number is unbalanced, but this is not a quantitative analysis of the variation. By using the integral intensity DAPI calculations that can be made in the cell browser software, we can calculate something called the DNA index. By definition, the DNA index of stromal cells equals 1. The tumor DNA index is then either less than 1 if the cells are hypodiploid and greater than 1 if the cells are hyperdiploid. By normalizing the whole exome data points with the DNA index, the whole exome data can be used to determine copy number variation. Using chromosome 12 as an example, we can see in the B allele plot, the blue graph, that the exomes within this chromosome are not balanced, but we can tell by how much. By quantitating the data, we can see that amplification of chromosome 12 has occurred 7 different times, and the amplification has occurred at different rates, from 3 copies of the exome up to 7 copies of the exome. In conclusion, we have 2 major workflows for analysis of single cells and FFPE samples. For single cells, we can offer you WGA for use with our downstream low-pass genome barcoding kit and cancer hotspot analysis. We also offer a whole transcriptome amplification kit for downstream RNA applications. The whole transcriptome analysis WTA kit is a sister kit to our WGA kit and was developed from technology licensed from Dr. Christoph Klein from the University of Regensburg in Germany. The Ampli-1 WTA kit reverse transcribes and amplifies mRNA from unfixed live single cells and groups of up to 100 cells. You can expect 2 to 3 micrograms of cDNA to be produced from a single cell. The amount of cDNA produced depends on the type of cell recovered and the cell cycle state of the cell upon recovery. The WTA kit also has a companion QC kit, which is also a multiplex PCR, which amplifies amplicons of different lengths. However, unlike the WGA kit, the number of bands that are produced from this PCR does not reflect the quality of WTA. You are looking for at least 1 PCR band to assure that the WTA was successful. The amplicons are located within housekeeping genes that not every cell that you isolate might express. For example, peripheral blood lymphocytes, PBLs, frequently do not express GAPDH, one of the QC markers. So if WTA was performed on these cells, the maximum number of bands that could be produced from this QC-PCR reaction is 2. For FFPE, we again offer you WGA for downstream low-pass genome barcoding, cancer hotspot analysis, and whole exome library preparation for whole exome sequencing. In April 2016, at the annual meeting of the American Association for Cancer Research, Menarini Silicone Biosystems announced the DEPArray NXT digital cell sorting system, the latest evolution of its flagship technology for precise isolation of rare cells from heterogeneous samples. The DEPArray NXT builds on leading-edge technology to reach the highest level of precision and resolution in separating pure single cells or group of cells for clinical oncology research applications. Lighter and more compact, the DEPArray NXT is a benchtop instrument that can fit in any lab space and allows one to obtain a pure cell preparation in a short time and with reduced operator interaction. It includes an internal barcode reader for sample-positive identification and unambiguous operator identification, automatic report generation, and advanced tools for image analysis. DEPArray NXT includes new software for image acquisition, which enables real-time visualization and interaction with all cell populations, and includes an option to preset target selection criteria, which reduces the hands-on time of the operator. Enabling the simultaneous recovery of samples using a 96-well plate, the DEPArray NXT is now a more efficient instrument for validated workflow. If you're looking for a needle in a haystack, not only do we make the needles really easy to find, we can even recover the exact needle you are looking for.